Hi, I'm Dana Null. I worked with Ashton Ferguson, Michael Newman, and Markus Whiterock, and we studied the relationship between our sleeping patterns and insulin sensitivity. We titled our paper, Differential Effects of the Circadian System and Circadian Misalignment on Insulin Sensitivity and Insulin Secretion in Humans. Here's a QR code if you would like to go and look it up yourself.
So we would like to talk about the significance of this study. And this isn't a very small scale effect, it is pretty widespread, mainly because the majority of the world and the United States, just everywhere, is actually pretty inflicted with diabetes. It's a pretty common thing going on around the world, and it's even more common among the low-income families and people in the United States and, definitely third world countries, because they don't have the same medical knowledge and equipment to be able to properly prevent themselves from getting type 2 diabetes or treat it. There are many other people who are at risk for type 2 diabetes, and our study definitely tries to focus on helping them out as well. And we mentioned that earlier we're going to be working with the circadian rhythm, and this kind of includes people who aren't getting the sleep they need, mainly the sleep deprived, such as people who are jet lagged, people who work night shift quite frequently. And we're also taking a look at the elderly as well, simply because their body isn't functioning as much as it should. And diabetes is a pretty big thing, and it has its own dangers. Amputation can be a lifelong and life-changing after effect of diabetes if it goes untreated. And definitely there's something called ketoacidosis, meaning that the blood acidity will rise in your blood and will throw off your body a lot and kill you pretty fast, actually.
Well, now that we have understood the why this research is being done, let's understand a little bit of the how on the circadian rhythm. The circadian rhythm is activated via light or the absence of light, darkness, which then sends signals through the retinal hypothalamic tract all the way to the suprachiasmatic nucleus, which is located in the hypothalamus of the brain. That then sends signals to many parts of the body which are involved in the process of eating, how it's digested, the metabolic processes of those digested enzymes, our mood, so depending if we're hangry, if we're happy, if we're depressed a bit also, our cognitive function, if we're able to think clearly and learn well, our sleeping, and we will be focusing on the normal day-night cycle, which will be known as aligned rhythm, and we'll be focusing on the night, which will be people awake and asleep during the day. That will be referred to as the misaligned. And this is very important for this study here.
So in order to better understand or fully understand our study and how it deals with circadian rhythm and diabetes and insulin and all that, it might be helpful to do a quick review of the pancreas, insulin, and how these things work. So to begin, we're going to have over here our glucose come into the bloodstream, and our very anatomically correct portrayal of the bloodstream and pancreas and other essential organs, as well as we have up here our key. So we have our glucose coming into the bloodstream through a GLUT2 channel into a beta cell inside the pancreas. What this does is the beta cell will detect the glucose and in response will secrete insulin into the bloodstream. Insulin and glucose will travel down the bloodstream to our other tissues and organs. For example, it comes to skeletal muscle or adipose tissue that is represented here by this block. Insulin will come in right here and attach this insulin receptor. Now what's important about this is that inside skeletal muscle and adipose are these GLUT4 channels. These are essential for glucose to come into the cell. Without it, glucose cannot get into the cell, being a polar molecule, cannot cross the cell membrane. But with the help of insulin, GLUT4 will come from within the cell to the cell membrane, as shown here. This allows glucose to enter the skeletal muscle, where it will be metabolized and used. In adipose, it will be stored. Those are processes that, there's some varied processes there between skeletal muscle and adipose tissue that we'll not delve into at the moment. Glucose will also come down the bloodstream and into the liver. Now, insulin isn't required for glucose to enter the liver, but what insulin is required for within the liver is to make glucose into glycogen, where it will be stored and used for use later during fasting periods. Now, diabetes, there's a breakdown in the system somewhere. In type 1 diabetes, that's when the pancreas fails to create this insulin in response to glucose. We're more interested, or rather, we see a high prevalence of type 2 diabetes in night shift workers, and that has to do more with the responsiveness of our tissues to insulin. That's represented here in type 2 diabetes. Insulin is still created by the pancreas, but the tissues don't really react to it. Glucose stays out here. It's not getting into the tissues where we need it. The liver might not be making as much glycogen. And that's where the issues are created with type 2 diabetes. And this is what we were looking for, any correlation with perhaps any circadian rhythm misalignment with either the pancreas being less productive or insulin being insulin sensitivity rather being reduced.
We studied the effect of the circadian rhythm on 14 healthy adults. All of our participants were non-smokers, did not take any medications, and did not do drugs. We put them through 2 8-day schedules where we controlled when they slept and what they ate. Before our participants came in, we required all of them to have 11 days of regular sleep where they got 8 hours each night. And the night before, we required them to sleep from 11 p.m. to 7 a.m. Here is our circadian alignment protocol. This is the typical sleep schedule for a healthy, normal adult. Each day, they were awake from 7 a.m. to 11 p.m. and we had each different days at different levels of light, which we will call lux. We had each of our participants sleep from 11 p.m. to 7 a.m. where they were exposed to 0 lux or no light. And every day, our participants had 3 meals, excluding the first and last day as these were short. Each meal was isocaloric and had about 50% carbohydrates, 30% fat, and 20% protein. And all of our participants were able to choose between 2 meals. For example, for breakfast, our participants enjoyed a dextrose solution we called glucola, a bagel with butter, and cereal with milk and sugar. But they could choose whether they wanted eggs and peanuts or turkey, sausage, and almonds. Our participants were also required to eat all of their food. From day 1 through 3, our lighting was set to 450 lux. This is the equivalent of a sunrise in clear skies. These were our control days to help everybody get on the same circadian rhythm. Day 4, we had the lighting to 4 lux. This was so that we could get an accurate low light melatonin test from each of our participants. Days 5 through 8 were our testing days. During these days, everyone was in 90 lux during wake hours, and this is typical of indoor lighting. We also had the morning at 450 lux for a short period to simulate a sunrise. We also tested the glucose levels in the plasma and the insulin concentrations of each of our participants around breakfast and dinner of day 5 and 7. We did this by testing 7 minutes before the meal for a fasting level, and then 10 minutes after starting their meal for an hour and a half every 10 minutes, followed by another hour and a half every 30 minutes of testing. This is our misalignment protocol. This protocol was designed to replicate the typical sleeping schedule of a night shift worker. To start this protocol, days 1 through 3 are the exact same as days 1 through 3 of the alignment protocol. It really changes at day 4. On day 4, we have the same level of lux at 4 lux, very low lighting, to get the low melatonin level for each of our participants. But we had them sleep from 3 p.m. to 7 p.m. instead of 11 p.m. to 7 a.m. to help them adjust to a new sleep schedule for the remainder of the test. After they slept on day 4, we kept them awake till 11 a.m. on day 5. And from then on, all of our participants slept from 11 a.m. to 7 p.m. For the rest of the protocol, our participants were exposed to 90 lux, which again is the lighting of an indoor room. We had mixed in a simulation of a sunrise at 450 lux in the morning. Our testing meals were the same, breakfast and dinner, days 5 and 7, with the same testing protocol.
So we'd like to talk about the method of how we were able to find our results. And so we use something called the oral minimal model. And this requires a 3-hour process of withdrawing blood. And so first we start off at, point 0 or just before the meal. We draw blood 7 minutes before. So then the meal will begin, and 10 minutes after the meal, we'll draw blood, or every 10 minutes after for 90 minutes. And after that portion is done, blood will be drawn for every 30 minutes up to 60 minutes. And here's a little timeline to show how that's kind of done. From the blood withdrawal process, we'll get our information and our values. Here's a diagram on how this whole situation kind of worked out. So our test subjects would eat the meal, and it would go to their gastrointestinal tract. And from the blood withdrawals, we'll be able to find the rate of appearance of the glucose in their bodies. And another value that we had to look out to eventually reach our conclusions and our data was the net hepatic glucose balance. And this is basically the idea that the liver helps regulate the glucose in the blood. But as you can see in the word, it's a balance. Not all the glucose is in the bloodstream or in the liver. There's a constant rate of it going back and forth. And then here we can also see there is a rate of disappearance, which the glucose is taken up by the body. And we'll eventually be able to find, insulin sensitivity, the remote insulin and just insulin in general.
We'll now move on to our results. Here's our first graph. On the left side here on the y-axis, this is our activity of our beta cells. On the x-axis, we have on the left biological morning. That's basically when the actual morning, like the AM, is happening in real time, not according to the body's own circadian rhythm, but the actual morning. And then biological evening on the right, same deal, the actual evening PM kind of deal. And here we're looking at our basal cell beta function, fasting beta cell function, and static beta cell function. Now, something interesting we found across all three of these was that in the biological morning, they were all more responsive or more active in the presence of glucose than they were in the biological evening. We have that represented here on the right with these two images. Again, in the morning, more insulin was produced by the participants' beta cells in the morning than they were in the evening. Misalignment is interesting because, as you will notice here, as compared to this last slide, there is no statistical significance in these differences here in misalignment between aligned and misaligned. That's what these two columns are for. Aligned is on the left and misaligned is on the right, these columns. And they were similar or the same throughout, whether aligned or misaligned. What was interesting about that is that we found that whether you're misaligned or aligned, the beta cells basically produce the same amount of insulin, as represented here. This person who would be aligned, being awake in the daytime and asleep during the night, made the same amount of insulin as our misaligned person down here, who would be awake more during the night than during the day, or be misaligned with the exterior day-night cycle. And finally, there's a behavioral cycle. Here we found that static beta cell function was the only one with statistical significance and difference. We found that in breakfast, when they've been eating breakfast, participants their beta cells were more active in creating insulin than they were in creating dinner, as represented in these slides, or these images over here on the right, using again our little cartoon insulins.
So here we're looking at our results of the insulin sensitivity. We're going to be focusing on simply the misaligned and the behavior cycle of our graphs. And so here we can see that first I'd like to go off by explaining what is insulin sensitivity. And that is the sensitivity of insulin to the blood glucose in the bloodstream. So in our misaligned, we can see that there is a pretty significant disparity along with the behavioral cycle. And our circadian rhythm, phase between the morning and day is pretty much unchanged. But what we can get from these two graphs right here is that as time goes on, our insulin sensitivity will decrease. So here we have our results for the total B cell function. And this refers to the glucose effectivity and how it secretes insulin and the rate that goes at. And so first we'll be taking a look at our circadian phase. And we can see that our total B cell function is higher in the morning than it is in the evening. Well, this makes sense mainly because our body, as it sleeps, will prime and get ready for the day. And so we'll have those B cell functions up and running, whereas later in the day, they're not going to be as active, mainly just due to our circadian rhythm. And we'd like to look at our behavior cycle next. And interestingly enough, we can see that for breakfast, our B cell function is low, as we didn't exactly expect that. And dinner, it was, much higher. And these were some of the results that we got from our test, the 3-hour processes of blood withdrawals and everything. And we can definitely say that our B cell function increases simply because we're eating a meal at a later time.
And now we are going to talk about one of the last graphs, which is involving disposition index. Disposition index is the effectivity of blood glucose and the amount of insulin secreted. For example, a lower disposition index would mean that there is less blood glucose entering the cells, meaning it's staying more in the bloodstream. And a greater disposition index would mean that more blood glucose is entering cells, which allows insulin to be secreted in a healthy amount. Now, with the circadian phase, which we will be looking at, we see biological morning, biological evening. Biological morning is when the sun rises, hence the term morning. Biological evening is when night arrives, hence evening. The biological morning had a higher disposition index than the biological evening. This lets us know that people in the morning, no matter if they had an aligned or misaligned sleep schedule, had higher disposition index, meaning, like I said before, insulin secreted in a healthy amount and blood glucose is entering cells, turning into insulin. We look at the behavioral cycle, in which breakfast had a higher disposition index than dinner. This helps prove the point involving the circadian phase that we just looked at, that morning had a higher disposition index, meaning insulin is secreted in a healthier amount than those in the evening or dinner time. And in terms of the misalignment for disposition index, there was not a statistical amount to prove that the aligned had a better disposition index than the misaligned, which is a little sad to see. I was expecting that to happen. And so with that, we'll head on over to the summary, in which the circadian rhythm, in terms of insulin sensitivity, had a significant increase for people who had an aligned living lifestyle than those who had a misaligned living lifestyle. Again, aligned circadian rhythm involved people who slept during the night and were awake during the day. And those who had a misaligned circadian rhythm stayed awake during the night and slept during the day. So, where do we go from here? Well, we wish that we can have a longer study, 1 month to be precise. If we have more, that'd be amazing, but a month would be sufficient, I feel, in order to see the long-term effects that misaligned or aligned sleeping has on someone. We'd also like to have a larger sample size, about 30 people, so that we can see the differences between each and every single person and how differences in body size, shape, and gender have a difference in terms of insulin secretion. We'd also like to see preset circadian rhythms, as in people who have already been living the lifestyles of the aligned circadian rhythm and the misaligned circadian rhythm, and see how that affects them when put into the study. We'd also want to see a blind study in which the subjects wouldn't be able to tell if they are involved in the aligned or misaligned study. And we also want to see how less sleep affects each person involved in the study, and how that can decrease or increase the amount of insulin they are secreting. And with that, if you have any questions, direct them to Jason Shaw at huntj@byui.edu. Yes, this is for real. If you have any questions, send it to them. They love answering questions. Don't forget to check that link out for the research paper we have in that corner over there. And with that, I bid you all adieu.